started. Thanks for joining us for a Scientific Computing and Imaging Institute Distinguished Lecture. We're very pleased to have Gene Myers here for our first Distinguished Lecture of the semester. I think Gene is well known to many of you. He's a group leader currently at Janelia. However, I was told this morning that that's a short-term proposition and that he's going to be a director of a Max Planck Institute in Dresden very soon, so building a new group there. Gene is probably most famous for his algorithm development of BLAST and bioinformatics, so for genome shotgun sequencing protocols. He was at cellular and helped develop the fly, human, and mouse genomes there over a 3-year period. He has a long list of awards, the Millennium Achievement Award, the ACM Kanellakis Prize, he's a National Academy of Engineering, and many, many other awards. But I hope you'll enjoy his talk, and please join me in welcoming Gene Myers.
Thank you. So, yeah, hopefully this will be interesting at some level to y'all. But basically, after we had sequenced several genomes in around 2000, the obvious question is what did they encode and what was the outcome? We've been promised kind of the blueprint of life. And, after a couple of years, I really started to think about what's the most significant outcome of this project. I mean, we didn't know how genomes work. But once we had the sequences, what I realized is we have the ability to basically intervene on a systematic basis, right? In other words, we can go in and we can take anything that we think is a protein and we can instrument it so that it glows. Or we can take any stretch of the genome that we think turns something on or off, and we can put it in front of a fluorescent molecule and see what it turns on and off. So in other words, I think really more than anything else what the Genome Project has enabled is not systems biology, but systems genetics. Okay, and I'm going to give you a couple of examples of the kinds of things that happen. I mean, these things have happened and are happening more and more. So I think the first example was on the yeast genome, where O'Shea and Weissman basically took every one of the 6,000 putative genes in the yeast genome, and they basically instrumented those yeasts so that each one of those proteins would glow individually. And then they put them under microscopes and visualized them to see exactly where the proteins are expressed. Right? And that's very important, because if a protein isn't expressed in a particular part of the cell, then it can't be doing anything there. Another example is Tony Hyman and his colleague Echeverry, who basically used an RNAi technique to basically knock down every gene in a little worm, C. elegans, and then they looked to see what the effect of knocking that gene down was on the cell division, on the first cell division. So you get the idea? So the idea is that suddenly you couldn't do this until you had the sequences, and we didn't have the sequences until 2000. But now we have the ability to do these kinds of interventions, right, on a systematic basis. So I call that systems genetics, because it's not systems biology. It's a systematic exploration of the gene. And another, oops, excuse me, I'm a bad driver. And then another example that you'll see a little bit later is with my colleague Jerry Rubin. We're basically taking every promoter or region that we think controls the turning on a gene to look at and get control of functionally related neurons. And I'll tell you a lot more about that. But the interesting thing that I saw about these kinds of explorations, which kind of appealed to me as a big biology, high throughput kind of guy, was that the outputs are all images. They're all CCD captures, right, of images or time-lapse series under microscopes. And that nobody was really thinking about how to interpret this stuff. In fact, I was appalled. I talked with Erin O'Shea and said, well, what did you do? And she said, well, we looked at them. I looked at 6,000 of these movies. I said, well, did you quantitate them? Did you, no, nothing, It's like, and I just keep thinking, what could they have gotten, right, if they had worked a little bit harder. So basically, I didn't really want to, I'm a sequence guy. I'm a traditional discrete mathematics guy. But I decided to kind of join your party and get into image analysis because I thought, mostly because I feel like that's where the wave is. in terms of the discovery and in terms of what I think is going to be really cool, high throughput genomics in the future. So let me just tell you a little bit of what I discovered in the 5 years that I've been doing this. So I would, just to give it a little bit of shape, I would say that people think about 3 levels of image problems. They think about problems that are inside cells, and those seem to involve one set of imaging conditions. So you're looking at how cells divide, how they move material around. Then at the next level, you look at collections of cells, so you kind of come out to 20x, and you look at nervous systems, you look at developing embryos, you look at collections of cells, and that seems to be another suite of problems to facing the informatician. And then finally, if you're going to do these interventions, oftentimes you want to know what's wrong with the critter. Like if you knock it down or you knock it up, what's happening? So you want to watch flies fly and worms crawl and mice whisker. Okay, so you want to actually look at phenotype, right? But the phenotype is an imaging phenotype, right? Okay, so, and then just, if you've never been in this arena, the thing that's interesting about microscopy is that generally SNR is pretty poor. Okay, it's, you're happy when it's 8. Okay, it's not like video. Okay, there's a lot of noise, a lot of background noise. Also, you're stuck. Especially with light microscopy, you can't go below the wavelength of light divided by 2, right? Nyquist's limit. So your resolution is limited. Okay, and the other thing that you have to be careful about is that you don't want to, if you're going to foray into this, I would just warn you not to get into this unless you're sure that this is going to be a high throughput deal, or if this is going to be an assay that's worth your spending a year or 2 of writing code for, okay, and thinking hard about doing mathematics. You want to make sure the problem's there to stay. Because I don't know about the rest of you, but I oftentimes have people come by with me with problems like it's one-off. It's like 1 or 2 days, and I say thanks, but I send them somewhere else. So, and the interesting thing though is when you find one of these puppies, what's cooler is that it makes us more important. It turns out that the computation is oftentimes the bottleneck, okay? All right, and I can, for example, for those of you guys who are working on connectomics, you're doing the EM thing. It's literally, you can collect all the EM imagery you want for a volume of fly's brain, but then it's going to take you 300 man-years to manually curate it at current performance levels, right? It's the informatics, and it's the level of accuracy. And oh yeah, I forgot that. I'm sorry, I'm not driving this thing very well. The important point is that you need to perform. Forget it. You need to perform at levels that are way beyond, like not 95%. You need to perform at 99.999, okay, accuracies. For some of these things. So very high levels of performance, which I think is interesting for people that really want to solve problems instead of graze through them. So what I'm going to do is I'm going to show you kind of an example of kind of forays that I've taken into each of these 3 levels. All right, and then I'll really cut loose and show you some stuff. I hope it's fun. So just to make sure you understand, this is an example of what you can see when you look inside of an individual cell. So this is, for example, so these are pictures of a cell getting ready to divide. This is the first cell division of a C. elegans. So it's the very first one, the first division in the developing C. elegans. And they're at different time scales. So this one is focused in on a very narrow time scale where there are 2 centrosomes that are set up. What just happened? Oh, I don't know. I don't think can I take this thing out and drive manually? Sure. Sorry, guys. Okay. And now I'm just going to get a laser pointer. So you have 2 centrosomes. The DNA, you can't see the DNA because it's not staying. What's staying here is EB1, which is the growing tips of tubulin fibers. And you can see all the fibers growing outward from this centrosome. They grow towards the cortex, where they're going to latch on and get pulled by motors to pull these 2 things apart. And then they're latching onto the kinetochores that are holding the DNA in the middle, which is creating a mitotic spindle, and they're going to separate. And so you're basically, if a cell's going to divide successfully, these tubulin fibers obviously have to be growing correctly. Here's another image in which what's stained is gamma-tubulin, which simply just stains just the centrosome. And you can see them set up here, and then you can see the cells. So this covers a larger arc of time. In fact, that was the nucleus. The 2 nuclei divide, basically disappearing as these centrosomes. So see the nuclei, they disappear, the DNA gets set up in the middle, these things start to pull apart, and the cell starts to cleave. And then finally, the worm needs to know where its head and its tail is going to be, and so there are two proteins, part 2 and part 6, which have been identified to set up that gradient, and you can see very clearly the fact that one is on the left and the other is on the right. Okay, and you can see them set up in the two frameworks. So you get the idea. I mean, this was the thing that kind of ignited me, was I realized, holy crap, I can look at a microscopic image and I can see all this stuff. I can see incredible details, actually, details that I didn't, as an informatician, I didn't even realize were there. I mean, I'm so naive. Okay, but not anymore. Now I'm totally into it. So, as you'll see. So, for example, what could we do with this thing? So Tony Hyman did these knockdowns, and he knows that 600 genes are implicated in mitosis and cell division. Any one of those 600 genes is gone, cell doesn't divide right. Something's wrong. Okay, so the idea is what we're going to do is what we're setting up to do is we're going to actually go back and take those 600 genes and mess with them. But for each one, after we've messed with it, we're going to take a look and see what's wrong. So we're going to build software that quantitatively extracts information about what's happening with the microtubules, what's happening with polarity, what's happening with centrosome formation. So we're going to try to do it right, okay? We're going to try to do a Shays thing right. So I already showed you that there are a bunch of different, so these are just things that if you attach GFPs to them will highlight various aspects of cell division that you want to see, okay? You want to see them go right. Okay, and so the idea is to literally take assays like this, so here are 3 of them, okay, and turn them consistently into good information. And this is where the computer's good. The human beings are really good at telling no, one of the centrosomes is gone, or holy cow, there are no microtubules. But human beings can't tell that the microtubules slow down by 20%, okay, or that the microtubules aren't necessarily reaching all the way to the cortex, right? Or, right? So human beings are terrific at qualitative things, but we really want to understand, because some of these 600 knockdowns are very subtle in their phenotype, and we expect that really the change, even though we know that something's wrong with the cell, he wasn't able to identify what was wrong, okay? And we're hoping that when we do this thing that we'll have a much better understanding of what's causing what in terms of the black box models. Make sense? Yeah, okay. Okay, good. So one of I think the most impressive ones that we've done so far is here we're watching 2 centrosomes, but not just through the first division, but through the next one, and the one after that, and the one after that, I think, in some cases. And we're able to basically be perfect up to 8 cells, and we start to have some errors at 16 cells, but very benign errors, as I'll show you. And I guess since this is an informatic crowd, I'm going to I don't think I can teach you guys anything about image analysis, but I can maybe impart some ideas about how we like to solve these difficult problems that you might find useful. So most of the time people develop heuristics, right? They develop, and then they report on an ROC curve for their heuristic how good it is. But the way I like to think about it is that, and then they pick this point and they say, well, this is it. I'm going to run it there, right? But there's another way to use these heuristics, because for a lot of them, you can find an operating point down here, right, where basically everything it predicts is true, okay, but it doesn't predict everything that's true. But nothing it predicts is false, right? So usually you can generate heuristics where the ROC curve actually touches the top and the bottom of the curve. And so you think about it, if you operate a heuristic down here, or you figure out how to operate a heuristic down here, and you get stuff that you know is true. You don't get all the truth, but you know you're inside the truth. Another interesting point is to operate all the way over here, okay? And if you operate all the way over here, then you're guaranteed that everything you predicted, all the true stuff is been predicted, but you got a lot of false stuff. So it's actually the black area. But if you take the complement of the black area, right, everything that's not reported, you know that that's definitely false and can be eliminated from the problem. Okay, so what you're doing is you're sandwiching the truth. Okay, so this is the truth sandwich. Okay, and the idea the way to solve these problems, for some of these problems, they're so hard that you're never going to develop one single computation that's going to have that curve hug way up in that corner and give you that 99.99 that you need. Just no way. So don't be so greedy. So do a bunch of these, okay? So, like, see, because after you've done so run a heuristic, where you got this characteristic, and once you're here, now you've got something to deal with. You've got, first of all, you've got less stuff you've got to compute on, because you've got rid of a lot of stuff. And you've got a subset of the truth that you can learn on if you want to do some machine learning or whatever, right? And you've also got a subset of truth that you know you can use to extrapolate outwards. So you can develop a series of such heuristics, okay? So it's very vague. It's not like telling you divide and conquer something. But I just wanted to kind of convey to you this idea because, frankly, if you go back and look at my assembler for the human genome, it's one of these, okay? It's a true sandwich, all right? So I realized that all I've been doing for the last 15 years of my career is solving problems with true sandwiches, okay? They're delicious. Okay, so tracking centrosomes. So, like, for example, everybody who does image analysis knows that the first thing you do is you run a Gaussian or Mexican hat filter to detect everything that looks like a spot, right? Okay, so you do that very conservatively. You over-predict the spots, right? And you get a whole bunch of putative spots. So what you've done is you've eliminated a lot of field of view. So the first thing you've done is you've reduced the complexity of the scene. Now you have to deal with 10,000 spots instead of, 100 million pixels, okay? Good. Good start, right? Then, now here's the important part. Now you want to find the tracks of those centrosomes in there. Don't go too greedy. What we do is we find tracks, we find subtracks. We don't find all the tracks. We find tracks on which we are willing to bet the bank. Okay, and that's always the part that's really kind of the coolest. So without really giving you an algorithm and spending a half hour droning on, I'll just say that what we did basically was a sensitivity analysis. We built an algorithm that had a number of parameters about how to connect things, and we ran it at different parameters, and we kept a core of it that stayed the same no matter how we turned the knobs, right? Sensitivity analysis, right? Those guys we bet the bank on, and sure enough, they're good, okay? See, but now, once you have the tracks, what do you know you got to do? So here's the core, right? So the spots are nice and bright, they're real clear, okay? What you do is now you extend the truth. I'm really having trouble driving today, sorry. Now what you do is you learn some statistics. We do actually learn the statistics of the movement of the things that are true. And then we use those statistics to basically extrapolate forward and backward in time because we know that they have to be connected to those things. Okay? So we're extrapolating from things that we know to be true. And when we do that, we get really pretty phenomenal performance. Like, I mean, look at those guys. Okay? All right? So the whole reason we did this was we wanted to do some interesting science, which is that it appears that the centrosomes get smaller and smaller with each successive division. Just appears. You just kind of look at them. You can see each time they're getting smaller. You go, maybe there's only so much material to make the centrosome, and it's not getting replaced, and so they're getting smaller and smaller. So the first thing is we had to quantitate this very accurately. And we spent quite a bit of time getting that right. You have to actually compensate for the PSF of the microscope. So you have to pay attention to details because it didn't look right at first. But indeed, it turns out that basically the volume is conserved. The total volume of centrosomal material is conserved, which says, gee, there must be somebody that's limiting the size of the centrosome, right? And then basically, just to cut the short story, because there's a lot of stuff I want to do, we basically did 153 conditions, 1,400 embryos, okay? So even though we spent a lot of time on this one assay, we ran it on a lot of images, okay? So it was worth doing, because without doing it, we wouldn't have been able to conclusively conclude that SPD2 is the mystery agent. In other words, there are other agents that are important in making a centrosome. We know they're important, but it's the fact that there's no more nor less of this stuff being made in those first divisions that's actually limiting what's happening. So this is actually a genetic explanation for the sizes of something, okay, for what limits the size of something. And so we got a nice piece of science out there, occurrence biology. Okay, so let me move to our first work on cellular collections. Where together with Stuart Kim, what we did is we took an L1 worm, which is easy to stage because it's just hatched, and we stained every nucleus with DAPI. That's easy to do. We basically made the muscle cells. There are 82 of these muscle cells that we stained with GFP, and they kind of form a line along the body wall. And then the idea is what we want to do is this thing has 558 cells. And what we want to be able to do is basically put a worm under there, kind of with a DAPI stain, look at it, compute how much, look at some protein of interest. So we're gonna stain some protein that we're investigating with the third dye, cherry, which is red. And we want to look and we want to say, gee, that guy's expressing this much cherry and that guy, and to know which cells those are. So for you that aren't biologists, the thing that's really fascinating about this guy is that every single one of them has 558 cells, and they all have a fixed identity. So if you want to do cell-by-cell genotyping, you can do it, right? As long as you know what the cells are, right? Like there's a big fat one here that's called, I think, M. Okay, everybody, every novice can identify M. There's a whole bunch of crowded ones in the head ring, which are very hard. But the point is that every cell has a unique identity, and it's always the same cell, right? So you can actually build a expression atlas, right, of every single cell in the organism. Right? You can know how much of each protein is being expressed in each kind of cell. And we even know how those cells are developed. In other words, the lineage is fixed. This is what Brenner and Salster got the Nobel for, figuring out that the actual series of cell divisions that lead to these 558 is fixed. So this organism has some nice kind of unique properties, right? So that's what we wanted to do. So the first thing we have to do is we have to straighten out this poor worm, as you can see below, because they don't cooperate, they don't lay straight. Okay, and then we have to segment it. We worked pretty hard at that. And then the harder thing, now take a look, I'm going to show you this. Notice that those are the muscle cells. See how they form four beautiful little axes? It'll turn out to be very useful to the story in a minute, okay? We call it a fiducial. It's an extra channel that's meant to help us do a better job of naming all the cells. And you'll see that it's very important. So, you stain the thing, you register it, you segment it, we kind of, and we segment it. And now we have a problem. We want to know, we have 558 cells, we want to know the name of every single one. Oops, who's going to tell us that? Okay, well, you can go to this big book and you can look at pictures, and they got pictures where they have them all labeled and stuff, okay, but we weren't good. So we were lucky enough to get a great postdoc in Kim's lab to actually sit down, Stuart Kim's lab, to actually sit down and label 50 of these things. But in order for him to do that, we had to create an entire user interface, okay? So our naive little project, which is just going to take us a year or two, okay, turned into 3 or 4, okay, because of things like this, okay? So anyway, we built this tool, which we managed to suck another paper out of it, where it allows you to basically kind of annotate these things and curate them. And the net effect is at the end, if you think about it, I mean, as a computer scientist would say, well, now you've got a ground truth training data set, right? You've got an actual, but inside, from a biologist point of view, you've got an atlas. We've actually got like 15 stats, which we kind of register to each other, and we know the identity of all of them. So we can actually give you physical coordinates for all the cells, which nobody's ever been able to do before. And I don't think it's a big deal, but I find it kind of interesting that it's 2012 and most of the stuff is still in pictures and books, yet we can actually take images of everything, of all these things. Why don't we have computationally available models of everything, okay? Well, as far as I know, this is the first one, okay? And it's interesting because we know things, like we know about from registering, we know how much the cells vary. So it's been well known that it's stereotypic. So, and indeed, look, we just kind of plotted the variation that we saw in the location. So we actually have the distribution, how invariant is the position of that cell. And you can see they're highly invariant, about a half a nucleus width on average, okay? So but that's reconfirming stuff. But the real point of doing that wasn't to have the atlas. That was still, remember, the goal is to be able to do single cell expression, right? So what we did then is we worked on basically, now that we have a computationally available map, okay? The goal now is when a new guy comes in, let's name all the cells, okay? And unfortunately, you can't just do it by kind of like juxtaposition or one-to-one. Stuff is horrible. So the most sophisticated thing we could do, where we actually used invariance, we made sure that, if this guy was in front of that guy in all 15, then in our answer it was in front of that guy. That helped a bit. That got us like an extra 10%, but it only got us to 80. We're like, ooh. And I'm not sure you could do better. But anyway, but the point is with the fiducial, doing the same thing and just doing things relative to the fiducial, suddenly it goes to 95%. Okay? And so this is typical. This is 95%, where the green ones are the ones that are wrong. And at that point, I think we were exhausted. Okay, this was meant to be just like a 1 or 2 year project, and it's like 4 or 5 years later, and we're all like, oh my God, okay, let's take a break. So we've taken a break, but what's cool is I think we're getting ready to do this one again. Okay, we're ready to take another run at it. We're reinvigorated because I mean, if you think about it, we got to nature methods and the cell paper, and we can actually like look at the tail part. We can't look at the head, but in the tail, if you're interested in kind of like aging, which is what Stuart's doing, it's good enough for that. So you can already do useful biology with what we produced, but it still isn't, as a technology developer, it's not, I think we can do better. And the real problem is, one of the biggest problems is, if you think about it, is that we segment and then we label. And there's an error rate here, and there's an error rate there, and if you multiply them, okay, you get 92%, right? And in fact, we achieve 88 because of, right, they amplify each other. So the reason we're only getting 88% accuracy overall is because of this fact that we broke those two things into into 2 different stages. So what's the obvious thing? Well, you want to do them in one step, right? Okay. But there's another thing also, which I'm going to explain to you, which is now that atlas was only over 358 cells, not over all 558. Why was it over 358 cells? Because that's all that Xiao could do. Okay, and to me this was very surprising, because I thought every C. elegans biologist could sit there and label them all up, and they all read the book from cover to cover. But no, okay, I think John Salston's the only guy in the world, okay, who can label them all. If you're a neurobiologist, you can label the ones in the head. I can get Corey Barkman to do the ones in the head. I can go find people that can do their subset. But somebody who knows the whole one, no, John, that's it, I think. Okay, so if you think about it, this is actually an important technological project, I realized. It actually made me excited, because really, the sub-name for this project is the Salston in a box. Okay, this is what we're really going for, right? It's the Salston in a box. It's important. And Xiao, bless his soul, actually, after some encouragement by yours truly, has actually managed to label all the cells, except for 12. So Xiao is about to become the second person in the world who can do them all. Okay, but we're actually going to have an atlas of all the cells. Notice that's different. We had a partial atlas. Now we're going to have an atlas of all the cells, and we can actually, another cool thing is we can actually do this on board a microscope. Okay, that's right. Start dreaming bigger. Okay, there's no end to our ambition. Okay, and we want to segment and register simultaneously, right? Which is kind of cool, problem domain, right? And the other, so I think between these two things, that in this next rev that we're working on, we're really going to be doing much, much better. Because we have a complete atlas, because when you look at like what were the mistakes that we were making, most of the mistakes we were making is there was some guy that was nearby that was completely innocent, but it wasn't one of the 358. And we pointed at it. And we got that one wrong, right? So a lot of our errors were benign. I mean, we were indeed producing, and we were just going off the partial map. Because so we had the map into, right? But if we have to do a one-to-one onto, right, that immediately is a much harder constraint, right? Which will help. And then we're going to segment. We're going to do template-based segmentation, right? With springs, it's fun. Okay. And so we think that we're really going to get to this goal, right, of being able to use single-cell medium throughput gene expression. I think it's going to be ongoing, and if my group isn't the one that does this, I think somebody's going to do this at some point. But we're into Rev 2, and like I said, and the cool thing is that if you can actually do this and you can get it on a microscope, then you can put your worm on a microscope, you can zoom through it, right? The microscope will run our software, will quickly will know all the cells. The neurobiologist who wants to knock out a couple of neurons, he can basically just put the worm there, type in the names of the 2 cells he wants to ablate, and walk away. Okay, and it's going to be much more efficient than him trying to find that thing and then hit it. Involve many fewer photons and basically save a huge amount of human time. Okay, so one example of whiskers. I'm pretty good. Whiskers is a phenotype of looking at a behavioral phenotype. Is we actually did a whisker. We haven't done much of this work. This is the only one we've done, but this one was kind of fun. So what you were looking at is a set of whiskers montaged over time. You basically got a little mouse who's got a probe into the barrel cortex that's controlling a whisker, And he's also got a tube that gives him a water reward when he does the right thing. And what happens is a post gets lowered. And the mouse has to learn that when the post comes down in a particular position, in this case forward, that it's going to get a reward. And when it's in another position, it won't get a reward. And then you observe whether it licks or not, right? So it has to learn. And we can actually watch what's happening neurologically while it's doing this. So our job, and the imagery is produced by a high-speed camera that's doing 1,000 frames a second, okay? That's basically backlit, so the light's from above and you're looking at kind of backlit whiskers, okay? And you're looking at a row of these whiskers, okay? Each experiment is 5 seconds, okay? So that's 5,000 frames. And then they do like typically 200 to 300 experiments a day. It's not unusual, trials, okay? And then you get these beautiful recordings, and the neurobiologists are having a lot of fun, okay, looking at this stuff. But the problem when they came to me is they couldn't even process one of these films, let alone there are 300. They were overflowing the disk. Svoboda's lab was at 200 terabytes and growing. Jerry came to me and said, "You got to do something. They're gonna bankrupt us with disk space if we don't do something with these movies." So we got into it. It wasn't really my choice, but I was trying to be a good guy. So this looks pretty innocent. You go nuts. Come on, this is easy. It's trivial, right? But no, it's actually not, because you can see there are things happen every once in a while. A whisker will flip over the post. There'll be a puff of air sometimes as an inducement, and the whiskers all go back. And sometimes these guys cross, okay? And they move pretty good, okay? Sometimes they come back and they're a little bit hard to tell from these facial hairs, okay? Depending upon where they are in the scene. And it's not hard to do 99%. You know, just all the obvious stuff right off the shelf gives you 99%. The problem is that in order to make this efficient, if you want to have one mistake per movie, you have to do 99.997%, right? If you want one mistake per movie. Because you also have to track them, right? You have to keep track of their identity. So 99 is not really good enough. If you give me a 99% solution, then I have, I have like a huge number of errors, right? I can't remember. It's like 500 errors to fix, okay? That's per movie, and there are 300 of them a day. The technician's going to spend, the postdoc's going to spend more time in front of the computer than they are collecting data, right? So we wanted, and then I gave this to Nathan Clack. I gave him my 99% solution and said, okay, it's got to be 99.99, okay? And you can't write a paper until you get there. So I won't tell you how we did it. Well, maybe I should tell you a little bit. I mean, we used another truth sandwich, which is we did something really obvious, which is we made sure we knew how many whiskers were in the scene. How did we do that? We counted the number of whiskers in every frame, and we took the modal number, right? Then we realized another really simple thing, which is that every frame which has the correct number of whiskers in it is usually correct, is a correct segmentation. So we deemed every frame that had the correct number of whiskers to be correct, and everything else that had the incorrect number of whiskers to be incorrect, right? Which is what you're seeing here. We then trained a hidden markup machine to basically learn what all the correct transitions were in the regions that were. So basically, I mean, this doesn't have to be perfect, but it's enriched for the truth, right? Okay, so it's a little bit, I'm not quite doing the truth sandwich. A little bit of crap on the black area, not much stuff. But it's so pure that when we train the HHMI, the HHMI is good. And then what we do is we finish the solution by reaching basically back and forth, okay, in a greedy fashion, using the HMM score to complete the thing. And we, generally speaking, get a perfect result, okay? And basically, we've measured it over, I don't know, hundreds of thousands of whiskers, and it's 99.997. One error every 30,000. And that's tracking. That's not segmenting. That's segmenting and tracking, keeping track of the identity. Okay, so I think that raises an interesting kind of challenge for you guys, right? It's like the same one that the EM guys do that are doing the neuron connectivity. The algorithm has to operate at levels that, you know, are way beyond what you need to get into CVPR, okay? All right? 99.999 is the standard, I think, for EM. Okay, so let me get to some, so that was kind of the survey. Let me tell you a little bit about what we're doing with fly brains, and then let me tell you about this cool microscope. that we're building that can image an entire mouse brain in less than a week. So if you've never seen a fly's brain before, and Jerry tells me this is an awful example, but hey, it's the one I found. It basically consists of 2 optic lobes, each which have 30,000 neurons in it, and it has a core hemisphere, which has about 20,000. And you know, I hope you guys are enjoying the biology stories a little bit. But you got to know a little bit about the problem to understand how to set it up. And you'll see that in a minute. So I'll teach you just a little biology real quick here. Okay, it's cool stuff, really. Okay, and the core hemisphere actually only has 20,000 neurons in it, but you notice it's bigger. And the reason is that these neurons are actually more complicated. You think about it, they should be. They're making decisions. They're doing things that are more complicated than the kind of the pixel parallel things that the optic lobe is doing. So if you stain the, so this is what you're looking at. Is you're looking at a stain where basically the green stuff is fat and the blue stuff is synapses. There's places where neurons are talking to each other. There's actually electrical connections going on. And the green stuff is actually from a glial cell, right? You have them in your body. You have big glial fascial sheaths, you know, on your neurons that go down your foot. But they also occur inside the brain. And they're basically, it's like the insulator of the coax cable, okay? It basically creates, and in fact, if an expert looks at this, they see compartments. They see kind of glially isolated or compartmentalized pockets. And in fact, a whole consortium sat down and decided that the first order, this brain is 50, each hemisphere of the core brain is 50 glial isolated compartments, and there they are. And they may not be biologically perfectly accurate. In other words, they may not be actually integration chambers per se, or units of neural processing, but they certainly give us a framework, right? At least they give us a terrain map, a frame of reference, right? Which is usually useful, actually, to the biologists, okay? So there it is in 3D. I'm sure you guys can do much better. Now, the other thing that you need to know, which was a complete surprise because we did not know this 5 years ago, is if you watch the fly brain develop from a single neuroblast, so basically that entire hemisphere gets built from 206 stem cells, neuroblasts, okay? And each one of those neuroblasts divides asymmetrically to form a neuroblast and something called a GMC, and I'm not sure why they called it a ganglion mother cell. But that ganglion mother cell then divides asymmetrically to create 2 neurons, okay? And then what happens? The neuroblast does it again, and again, and again, okay? As many times as it needs to. And each one of them does it about 100 times to make about 200 neurons, okay, to give you your brain. Okay, and the interesting thing is that these guys really actually know their identity. In other words, the red ones know they're red, and the yellow ones know they're yellow. So we talk about, instead of talking about A lineage, we talk about the A hemilineage and the B hemilineage. Okay, all right. So in the early stage, you make a little brain, and then later on, you make another brain, because you know flies 2 critters, right? It's a maggot for a while, and then it pupates, and it becomes a thing that flies. And you get about 200 neurons from neuroblast. So now here's the thing that we did not know, which is really amazing, because you think, wow, that's really special that worm that has 558 cells and they all doing exactly the same thing. No, my sense of it now is that stereotypy, which is the tendency for things to be in fixed places, occurs all over the place, especially in early development. Where it makes sense, right? In hindsight, it's like, oh, you're so stupid. It's like, yes, if you're going to make something and you want to plan, then where you put the first guys, it's important. Okay, so the genome would spend energy to put them in those places. So if you watch what happens from, say, take a single neuron out of hemilineage A, and suppose it goes to compartment A. Remember those glially isolated compartments, and it's basically going to integrate signal from the proximal arborization. And then it's a long conduit, it divides into 2, and then it basically creates primarily axonal arbors, where it then basically pushes signal into other neurons in the compartment that are integrating that signal. If you look at the other guys, if you look at its siblings, okay, you get the idea, right? They all do the same thing. They're all under genetic control. So it makes perfect sense, right? Because I mean, a fly's ready to be a fly as soon as it pupates, right? It doesn't come out and go, gee, you need to feed me for a while until I can figure out what to do, right? It knows how to mate, it knows what it likes. Its brain is ready to go. It's a fully functional right? It's a little FPGA, right? It's ready to compute the brain, you know, mentality, right? So it makes sense that it's completely, I mean, they're stochastic elements, right? The arbors are definitely stochastic entities, right? Fractal entities, but I believe that my bullshit theories that there's only a certain level of fractal programs, and those fractal programs are producing receptor fields. And so that this thing is, in some sense, so nature is basically modulating at the level of this gross circuitry, at the level of what these arbors, what the fractal patterns for these arbors are, and then there's one other set of things which is it decides which ones to arbor with, which is kind of a sniffing thing. I'm only gonna talk to you. Okay. So, but those are three orthogonal axes around which you can build a brain. Okay. At least a fly brain. But this is very exciting for us as people that want to kind of build a model of a fly brain, because it means that we can look at thousands of flies and know that they're all the same. Okay. They're all stereotypic. The fly's brain is bloody the same at this level, the level of drawing of this picture. So the first thing you want to know is just what's its hypergraph, what's the flow of information, right? Things get integrated here, and they get propagated there, and then somebody sets off and does another thing. So we actually believe that we're very close to actually having a map of basically all of the pathways, right, through that fly brain. So we'll know how information flows at the gross level. The other fascinating thing, by the way, that we found about these things is that so these three guys that put arbors in here, guess what? Those arbors don't intersect. They fill out. They basically, like, they'll take half of this compartment and they basically will fill it. So they've also got some kind of antisense thing, right? So they're just designed to basically integrate this volume, right? Or to enervate that volume, right? If they're transmitting the signal. So it's really pretty, I mean, I thought this was pretty cool. And this is all stuff we didn't know 5 years ago. We didn't know how stereotypic the brain was. So we know, so we can solve certain problems. We can register these brains really well. You guys do the medical stuff, you know. It required some finesse to get the brains. We can trace individual neurons, but we can't. But if you light up a lot of stuff, we can't trace it. But we can light up sparsely labeled things, and that as I've indicated, that fly brain is highly stereotypic. So what we're going to do is we're actually going to try to build a model, really, of every neuron, of every single one of those 20,000 neurons, okay, and where they go using a hierarchical approach. So the first thing we do is we use a systems genetics approach, like I talked about. We basically take the fly genome, which we have, and then we take little 500 base pair pieces, or 3,000 base pair pieces, right, and we stick them in front of GFP and we see what parts of the brain they light up. So here's an example of that from Barrett Piper, Jerry's lab, where basically see these blue regions, these are regions of the genome that are upstream of this gene, or actually this one's interspersed with the gene. And you take those and you have them drive GFP, and this is what you get. And you can see that they're staining, that only a very small subset of the neurons, this is the VNC, by the way, the spinal column. You can see that very little sparse bits of this thing are actually being lit up. I mean, here, we've got a little oval there, okay? So we don't know which neurons. What we find is when we do this is 50% of we pre-screen and take regions upstream of things that we would suspect would be differentially expressed in the brain, okay? And then when we take these things, we made a guess. And then when we take these things, 50% of them are interesting, like this. This is very unique because every single one of us do it, okay? And 50% of them we just throw it away because it either lights up the whole brain or it doesn't light up anything, okay? But 50% is a great hit rate. Okay, and after 3 years of work, okay, and 3 years of imaging, we basically got all these flies. And then, as an informatician, you're like, well, what good is that? Okay. Well, what it is that I have reproducible access to a subset of the neurons. I mean, yes, it's a subset that it has perhaps no biological meaning because it's an arbitrary promoter, but I have reproducible access to that subset of neurons, right? And I can do things. I can knock them down, I can knock them up, I can build a map of all. We have 4,000 of these constructs that basically cover the brain and give us really refined access to subsets of neurons. So we can start to begin to just do black box experiments to figure out what goes wrong with the flies. And there's another whole team at Janelia that's basically like monitoring flies. They're watching them crawl and walk and jump over things and fly. And so there's a lot of that kind of phenotypic work that I'm talking about. So that's level one, and we're looking at this now, and I think we're really close to being able to give you that hypergraph, that 50-node hypergraph. But we can do more, and we're starting to do that, which is that now what we're doing is now that we basically got a molecular handle on all these guys, now what we're going to do is bring this sparseness, which we're going to sample individual neurons. So we're going to use what's called a Cre-recombinase. It's a genetic technique where we can kind of stochastically decide whether or not we're going to turn something on. So we'll take one of those things that lights up 100 neurons, and then we'll flip this thing in 2 or 3 of them, maybe, or maybe just one of them will get turned on. But we're going to do even more. We're going to use color to help us. So if you guys know the rainbow work, right? We actually going to have red, green, and blue ones that we're turning on with some probability. So we get neurons in different colors. And this is, and we're not doing it because we're trying to light up all the neurons. Notice it's very sparse in the optical. But what we want to be able to do is we want to be able to get as high a yield as possible. So for each stack, we want to get like 10, 15, 20 individually traceable neurons. So we use color to help us get more data per volume. And we think that if we do 10,000 stacks, right, we'll have a 5x sampling, right, at 10 neurons per stack. We'll have a 5x shotgunning, as it were, of the fly brain, right? So I'm back to my shotgun thing. Okay, here's another beautiful one. And you can even do more, because I mean, so this is all we're going for right now. So we're starting this thing in phase 2 now. This is cool, we're working on this stuff. But phase 3 is like, you know, once you've got every single one of the neurons individually mapped, then you can start to think about, well, do they interact? So if you had a good assay that told you whether or not 2 neurons were interacting, and there are some protocols that look like they may give us that information, you know that in order for 2 neurons to interact, they have to physically be in the same space. Well, we may not know exactly where these guys are, but we know to within a couple of microns. So we can be conservative, but notice we don't have to explore all n squared. We don't have to do 20,000 squared. We only need to do, for each one, we need to explore the ones that could potentially interact with. It's a much smaller number. So we're talking about maybe 100,000 interaction pairs, 200,000 interaction pairs. So we could actually even potentially get interaction information. So, you know, our team's part in all of this is, apart from kind of kibitzing on the overall game plan, is to basically build a software that makes all of this possible. And so these are the things we got to be good at. We got to have good software for registering fly brains. We got to be able to color separate the neurons. We've got to be able to trace them and make models of them. We've got to be able to tell what compartments are being innervated. I'm going to just jump ahead. I'm going to show you, you guys have done a lot of registration. We do landmark-based registration. I think the thing that's cool about what we do that's perhaps novel is rather, I mean, the most important thing in registering 2 things is knowing whether or not 2 points in space represent the same point, right? I mean, that's fundamentally it, right? Is to understand what that mapping is. So a lot of schemes have been devised. So what we did is we just said, what we're going to do is we're going to take 5 or 6 of those, and we're going to run all 5 or 6 of them, and we're only going to take as corresponding those points for which all 5 or 6 of them agree. It's sufficient concordance to say that we definitely got it, right? So we call that a confirmed landmark. And we can tell how good a registration is by just looking at how many confirmed landmarks we have. And in fact, that turns out to be a perfect figure of merit for how well the dissection was done. So I mean, it probably doesn't make any sense for like NMR brain stuff, right? But for, you remember, we got dissectors, right? And if they put a big forceps divot in one of their fly brains, that guy's out, okay? All right? And so we need to know that kind of stuff. And so here's the fascinating story, though, which is that we set this thing up with this figure of merit. We started giving them feedback reports, and it got better. It got a lot better, okay? The protocols got better, the dissectors got better because they started competing with each other for who could produce the highest numbers. Okay, so it went from basically, we used to be able to register 50% of the brains that came out of the pipeline. Now we register over 85%. I mean, you know, so now we got a real yield. The other thing is that, so this is 100, the other thing is that we really, I don't know about how you guys, but I'm really happy with this. This is like 100 different fly brains in which the neuropil has been aligned. And the fascinating thing to me is that you can see these sub-regions. These are subglomeruli. These are regions of concentration where a bunch of neurons are making a little focused kind of interaction bundle. They're called glomeruli. And so you cannot see these in an individual image. But when you take our registration of 100, you see them, which means that we're actually not, you know, there's kind of a point where things converge and there's a point where they diverge. We are not, we are in a convergent regime, right? And I will prove that by showing you the brain for 1,000. Okay, so notice the extra 0 and the fact that you can't tell anything else. Okay, and I swear I've not sleight of hand. That's a different stack. Okay, so we feel that we've solved that problem. You know, multicolor separation is not that interesting. I'm just going to keep going because I want to get to the scope. So we do that. We're doing that good. This one maybe is worth talking about for a minute. So neuron tracing, because it's a problem that's been studied to death and nobody does well. Okay, nobody. There's been a ton of papers and no adequate solutions. Okay, tons of papers, including us. I've produced inadequate solutions and written about them, I must say. I'm embarrassed to say. But I think we finally got it right with something that was not, you know, because most of the time you want to do like a complex manifold, or you want to look at derivatives of the signal, or and that's the kind of stuff that gets you into CDPR. But here's what you do: you want to basically, this technique boils down to, you can see that the information is punctuated, right? So notice that there are kind of the bridges out, all right? But it's really clear as a human being, you know how to follow it. So what do you want to do? Well, it's really simple. We first identify everything that's clearly foreground. Okay, so again, it's like the true sandwich thing. We don't try to get into the gray zone where the pixels we're not sure about. We just take the pixels that we just know they're so bright. They're 4 sigma above the background. Okay, they're foreground. They must be explained. Okay. Then what we do is we take a point. We just pick a seed in a nice bright spot, and then we say, okay, find me the geodesic distance, shortest distance to everything that's foreground. Shortest paths, right? Well-known algorithm, linear time. Okay? Then what we do is we say, okay, well, that's a little bit much. Let's take the subset of that, the sparsest subset of that, where if I put a sphere around the thing of some radius, I can pick the sphere, it explains the data. Okay? And when you do that and peel it back, ta-da. Okay? So it's, I'm a little bit embarrassed. It's not the world's most complicated algorithm. But it's really robust, and it really works on all the data that we've tried. Okay, so I don't know. So, I don't know if that's disappointing or exciting. Okay, so, because it's not very sophisticated, right? Oh, well. But it works. Oh, dang, it's really good.
And does it maybe teach you something about the way that the neurons actually, the way the brain is developing, and the neurons actually are structured?
Well, no, it's actually appealing to the fact. We're just trying to, you have an important prior piece of information. I said strong priors. You know that a neuron must be a tree. And you know that you have to explain all this. That's it. That's it. Right? We just find the sparsest explanation that gives us a tree that explains all the signal. Yeah, okay, let's move on. I want to show the microscope too, because I think there are a couple of people here who might find it interesting. So there's compartment analysis. We can do that. Okay. So suppose that we wanted to do a mouse brain. So suppose we wanted to do a really big volume. Okay. Well, and why do you want to do a really big volume? Well, here's a soma, here's a cell body in the mouse, and if you zoom out, it's in the middle there. Okay, if you can still see it, and if you zoom out some more, and the point of the matter is that neuron is actually sending its projections all the way through this material. So in other words, you can have a neuron, which is like 200 nanometers in diameter, travel 5 millimeters or 10 millimeters. So you have a microscopic object that you want to follow over, an astronomical distance, right? The only way you're going to do that is with a microscope at 63x, okay? So what we did is we said, well, can we build a microscope that will image that volume in anything near a reasonable amount of time? So you saw those coronal sections that took, to do one, just to do that one section took 80 hours of confocal time, okay? That one that we showed you, okay? So what we wanted to do is we wanted to build a microscope that was 100 times faster. So, here are the specs. I'm way late, so I'll just say, and so this is kind of interesting for me because I'm actually starting to do physics. We came up with an idea for a 2-photon microscope that's block facing. So the idea is that you'll basically just take the mouse brain, you'll embed it in agar, you'll clear it, so it's strictly in situ, you'll put it in a bath, and you'll walk away from it. And a week later, you'll come back and have the whole brain done. 4.2 trillion voxels in 6 days, okay? no hands, all right? And no registration issues because there's no sectioning. It's block face. You're going to get a perfectly registered grain, okay? Overlapping cuts. So there's an onboard microtome, and we're almost there. And in fact, I can show you imagery, okay? So, I don't know, I'm pretty, am I going down? Yeah. So, I'm really happy with that. I mean, we've got all the kinks out. This is moving at 8.6 million boxes per second. We did this slice, which is exactly the same volume as the thing I showed you. We did it in less than an hour. Okay, so we got a factor of 86. We didn't quite get 100. We're going 8.6. We're going 5 times slower than the PMT. Okay, so not much room to go faster, and you can see it's really beautifully clear. And so we're really happy with the optic path and with the speed. And I won't bore you with all the technical solutions. We've gone through all different parts of the sample, and really what remains for us is really the automation aspects. We've just got some. I'm kind of embarrassed because I was hoping to be there at this point, but we've had to do pretty sophisticated acquisition software because you're talking about hundreds of times, right? So so we have things like if there's something that's slightly too bright, and we don't know it because we haven't scanned the whole object. When the scope hits it, it turns off the PMT. Okay, that's not good, right? So we have little things like that to work out, and we can solve that, okay? But it's just like nobody's ever collected 20,000 stacks, right, with nobody around, right? So all we're almost, so you can see, I mean, we've got the optic path, we're almost there. I mean, I really think like another 3 months, and we're going to be able to do the entire mouse brain as I've outlined it. We're just down to instrumentation issues, and I'm very excited about that. So let me leave you with one last thing, because I am moving to Dresden, okay? And the reason I'm moving to Dresden is because I really believe in the following picture, which is that this is what I think biology is starting to look like more and more. It really depends. I mean, how well you can do as a biologist, it depends upon what can you look at? What can you observe? What's your technology? I mean, to the person with the highest technology and the best equipment go the spoils. Okay? All right? There's the actual experiment itself. So, I mean, this technology can't just kind of be happening in a vacuum. It has to be directed towards something, and that's it. Okay, that's the focal point. That's the biologist. But once that experiment's taken place, if we're talking about these microscopes and these things, then analysis is incredibly important. And I really want to do this. I want to be able to extract models of the neuron. And then I don't want to eliminate the last part of it, which is part that I actually have never been very strong on, which is that you need a theoretician to actually explain to you what it is, give you models that explain what it is that you've worked so hard to extract from your system, from your living system, right? So in Dresden, they're incredibly good molecular biologists, biophysicists, and theorists. And so I'm basically going to go and do the 2 things that I think I kind of know how to do, which is Bioimage Informatics, okay, and to basically work on building the optical engineering and the molecular reagents. Okay, I actually can't do this part, but we have people that can do that. But I think now that I've actually built one microscope, I'm pumped up to build some more. Okay, some ones that are really cool for this application. So I'm actually going to show you an image from actually a colleague that I worked with, Philip Keller, who's built what I think is the current best spin scope. Although I think we can build an even better one, and we're going to try. And here you're watching the beginning of the development of the fly wheel. So I'm going to be basically looking very much at the development of flies, worms, and zebrafish using this kind of technology, as well as looking within cells with much better resolution instruments, such as structured illumination, Bessel beam, 2-photon, if anybody knows what that combination of words means. Okay, but it's really gotten to the point where you really can. There's a beautiful set of techniques for intervening that are high skill techniques for making observations that you can't buy off the shelf. You can't go to Zeiss or Leica and get them. All right, you may 10 years from now, but the whole concept is that if you really want to answer your question, you build your microscope around the question. not the other way around. You don't buy something from Leica and then try to figure out how to retrofit it. You build a scope for the particular conditions. Like the scope that I built for the mouse brain, I don't know if it's good for anything else, okay? But I don't care, okay? I know that it's good for mouse brains, and I know somebody's going to want to use that. In fact, the group has already realized that they don't want to use the nanozoomer anymore. So they had a wide field instrument where they were just doing kind of wide field sectioning. My scope is basically just as fast, a little slightly slower per plane. But that's against wide field, by the way, right? And I'm doing basically a confocal scheme. And because I do block facing thing, there's no registration issues, no slices. So they want to use it because they're going to get a perfect model. They're just going to sample every 50 microns. But they'll be able to, in the same frame of time, get basically, they'll get, so unfortunately it's no employment for us because I'm getting rid of the registration problem, right? No registration required, okay? But it's really hard to register slices that are that big, okay? And so, you know, but I hadn't even thought of that use, okay? And so I'm really looking forward to going to Dresden, and I mean, I hope the society hasn't made a mistake in investing in me, but I'm going to be trying to do the very best I can with the image analysis that comes out of these kinds of scopes from looking at development. And we're also going to, a lot of the team is going to be building scopes. So it's going to be an interesting group. It's going to be physics, it's going to be instrumentation. We've got very good people at instrumentation and control, and then classic image analysis and information. So, and I guess I hope you'll forgive me. I'm just blatantly advertising. Okay, I'm building a team there. If you're adventurous, you want to have your like European adventures, check me out. Okay, thank you. Thanks.
I think we have some time for questions, as long as there's not a class in here. Okay, good. Yes, please.
Sorry, I went long.
That's fine.
So with respect to using the segments from the fly to light up different subsets of neurons and everything in the brain, as you look at tiles, do they tend to be simply additive, or are there any general rules that emerge? So, you know, if you have them all around the gene, you get, you know, a bunch of neurons lit up, take this one, the one next to it, you get half of them over here. Or
You mean in terms of the promoter construction?
Yeah. Do the different tiles add, or do they give you?
No, Sean Eddy's looking at that, trying to see if he can. I mean, basically, no, we're not really seeing. I mean, these are artificial promoter constructs, so they're just responding to some set of transcription factors in some artificial way, right? But reproducible, right? In the context of the cell. So, you know, I haven't spent a lot of time looking at them, but I haven't seen them. I know Sean has, and he hasn't kind of ever said anything like that. What he's doing basically is looking for enrichment. In other words, he looks for guys. So he takes all of the constructs, he looks for guys that are turning on the same things, but from different parts of the genome, and then he's saying, can I find promoter components here? Right? So it's a nice way of actually getting pieces of DNA that you know are binding to the same TF, right? Or same set of TFs, right? So it's more like that, I think. Yeah?
The questions, Farley?
So you mentioned how the brain has a template, and I was wondering
How the brain is what?
the brain is constructed based on some kind of a template of construction.
It's highly stereotypical, at least at the level of fly brain. The human brain is not, clearly, but
Right, so I say the fly brain, but even on the level of the fly brain, people study learning in the fly brain. So I was wondering if you could see any plasticity or changes in the
Yes.
brain.
well, you can have Pavlovian condition of fly. You have to be very patient, but yes, you can.
You can see it in your microscope, and you can see it in your using your
We have not been able, the only thing we did is we looked for sex differences. We looked at, is there a difference between males and females? And there most definitely is. There most definitely is. It's subtle. It's just like slight variations, but statistically significant variations in the routing and the sizes of things. But otherwise, still stereotypic. I mean, still the same structure. It just looks like the weights, the synaptic weights of things are different. And so at the growth scale, the wires are all in place. It's really what's being changed with in terms of the learning is just really the synaptic density, the formation of synapses, right?
Right.
That's what's happening when it's learning.
So is that a question of resolution? If somehow you had better resolution, you might be able to see this, or is that a question? What kind of technology would you need to see changes due to learning?
you'd have to look at the electrophysiology.
Okay.
You'd have to know. You actually have to look at the signal. You can't just look at anatomy. You have to know what. You'd have to be able to look at the spiking patterns. And right now, you have to patch clamp to get a reading that going to give you the information that you would want. There's the genetically encoded reporters are getting better, but they're still too slow for you to really see that. So there's a hope maybe in 5 or 10 years with improved chemistry that there'll be reporters, the genetic reporters that you can actually put in and you can observe with light what the firing patterns are. But you're going to have to look at the firing patterns because synapses are very small. They're at the limit. I mean, a synapse, it'll show up as a little punctate spot. It's 2 or 3 pixels at 63 amps. That's it. So you wouldn't be able to notice anything unusual about what's happening there. I mean, you might be able to look at the number or to be able to count them or to molecularly type them, but it's a little too far out. I'd be just unspeculating.
So apparently there's a class in here.
But
okay. Gene has about 20 more minutes, and maybe we can just move out into the hall for people to ask some more questions. Let's thank Gene again for that. Thank you, sir.
Okay.
Great. It was fun.
Sorry I went long, but I knew.